Hello, and welcome to this webinar titled Effect of Cell Culture Process Change from Fed Batch to Continuous on Productivity and Product Quality, hosted by Biopharma Asia Magazine and presented by Zang, Associate Director at Mersana Therapeutics, and John Bonham Carter, Product Line Lead of Cell Culture and Clarification Business at Repligen. My name is Stephen Edwards, and I'll be your moderator. Now please allow me to introduce our first presenter. Zang, PhD, is Associate Director of CMC at Mersana Therapeutics. Prior to Mersana, she was a Senior Process Engineer at Momenta Pharmaceuticals, where she led a group in establishing the continuous platform for process intensification. She has 6+ years of experience in pharmaceutical industry, and her expertise include biologic process development, process characterization, scale-up and scale-down, tech transfer, GMP, product management, and CMO management. Before Momenta, she was a scientist of late-stage process development in Sanofi Genzyme. Dr. Zang got her PhD degree in chemical engineering from The Ohio State University. I will now be handing over to our first presenter. Welcome, Dr. Zang.
Thanks, Steve, for the introduction. Good morning, everybody. Welcome to the webinar. So today, I'm going to talk about the effect of cell culture process change from fed batch to continuous on productivity and product quality. This is the outline for today's presentation. So first, I would like to give a brief introduction to process intensification, followed by manufacturing options, fed batch versus continuous. Then I will talk about the effect of process change from fed batch to continuous culture on productivity and product quality attributes. Also, the feasibility of modulating the PQAs in continuous culture is also evaluated in this presentation. And the last part is a summary for today's presentation. Let me start with process intensification. Process intensification is not a new concept in traditional industry. It was first mentioned in 1970s by Kleeman and Ramshaw. It describes the strategy of reducing the size of chemical plant needed to achieve a given production objective. Process intensification offers the potential to reduce footprint, cost, as well as environmental impact. At the same time, process intensification can also increase the production output, improve value and the product quality. So for current biopharmaceutical industry, the current process intensification approaches include high cell density backing, which can shorten the seed train duration, N-1 perfusion, and high cell density inoculation, which can reduce the manufacturing duration. Perfusion production, concentrated fed batch, continuous chromatography, as well as integrated continuous bioprocessing, which was proposed by Konstantin Konstantinov in 2010 at the 12th Cell Culture Engineering Conference. So several industries, including petrochemical, food, and chemical industry, they have gone through a manufacturing evolution from batch to continuous processing. But for biopharmaceutical industry, it is still in early stage. So currently, there are two continuous approvals. The first continuous approval is Orkambi for cystic fibrosis disease, manufactured in Vertex. It was approved by FDA and EMA in 2015. And the first batch to continuous process change approval is for Prozesta, manufactured in Janssen. It was approved in 2016 by FDA and 2017 in EMA. So there is growing interest from the industry, academia, or even from the regulatory agencies. So Dr. Saul Larry, the chair of the emerging technology team, and he noted in 2017, he said the transition is challenging but worthwhile transition. and the FDA is providing resources and information to help facilitate the transition. So far, there are more than therapeutic proteins produced in perfusion mode have been approved since 1990s. Among those approved therapeutic proteins in perfusion, more than 70% of the products are unstable products. For example, is recombinant enzymes and blood clotting factors. And only less than 70% are the stable products. In general, is monoclonal antibodies. So currently there are 6 commercially available antibodies produced in perfusion mode. Is Real Pro from Johnson & Johnson, Remicade, Stelara, Simponi, all the 4 from Johnson & Johnson. Simulect is from Novartis, and Compounds is from Zang. So basically, for the continuous manufacturing, the majority is still for the unstable products based on the summary. So as we all know, fed batch and continuous are the 2 dominant manufacturing modes for the industry scale production of therapeutic proteins. Both of the modes, they have their own clear advantages. For example, for fed batch, so its clear advantages including the process simplicity, because people have been working this for decades, and they have extensive experiences. And it's well-established high throughput scale-down model. For example, the Amber 250, a lot of like biopharmaceutical companies, they qualify those high throughput as their scale-down model. And also, fed batch can offer the better medium utilization, so lower costs. And also for fed batch, they have established and extensive experiences in process characterization and process validation. On the other side, for the continuous manufacturing, it also has a lot of very unique advantages. For example, it can reduce the footprint, which people keep talking about, and also it has a high volumetric productivity. Continuous can also offer the steady-state quality attributes. And also sometimes for the continuous, the development scale could be the same or similar to the manufacturing scale. So basically for this way, it can minimize the risk of scaling up. And also continuous can offer the streamlined process by integrating the downstream, upstream, all the way down to DP. And also it can lower the capital expense and operating expense. And also it has increased the flexibility in capability management. So which is the winner for both manufacturing modes? So for unstable protein products, the answer could be clear. Continuous will be the preferred option for the manufacturing. But for the stable products, for example, monoclonal antibody, so there's no clear answer which one will be the winner. As we know, the advances in cell line engineering and medium design, it has led to the significant increase in fed batch titer, so which is reported around 14 to 15 grams per liter by Genetic and the 18th Cell Culture Engineering Conference. So with this, fed batch may still be the major platform for the short term. And also Perfusion from Johnson, he noted in the 10th annual bioprocessing UK conference in 2013, Johnson, as we know, Johnson is the main perfusion player, and he noted for the commercially available monoclonal antibodies, they will still stick with the perfusion, but for the new clonal antibodies, so moving into clinical manufacturing, so they will move to fed batch platform. So for these slides actually summarize the case, the scenario analysis while looking at those 2 manufacturing modes, continuous and fed batch. As I mentioned earlier in the previous slides, for unstable proteins, such as enzymes, blood clotting factors, continuous will be the preferred platform. Another scenario for continuous is for low-producing cell line. For some of the old cell lines, for example, for the specific productivity lower than 10 picogram per liter per cell per day, so those ones, continuous could be a preferred platform. And also another case is for some cell lines, for example, the sp2 hybridoma cell lines, they may not grow well in fed batch. So in this case, continuous could be another preferred option. For novel stable products, the monoclonal antibodies, so both continuous and fed batch, they are good. There's no winner for that one. Regarding which one to choose, that may depend on the company strategy and also the preference. For us, for today's focus is we are talking about the stable products for the late stage, novel, legacy, or biosimilars. So for these products, regional challenging. As we know, for biological products, transitioning from fed batch to continuous may not yet exist. And also, there are few literature really talking about the challenges for this transition. So for today's presentation, so we're trying to evaluate the feasibility of process change from fed batch to continuous for the late stage, legacy, or biosimilars. So as we know, it's very challenging for this transition, since this significant process change may impact the product quality attributes, which may further impact the safety and efficacy of the products. So we know glycosylation is a very important property for the clonal antibodies. It may impact the efficacy and the in vivo life. For example, the high mannose may impact the ADCC or clearance rate. Co-alkylation and glycosylation may impact the ADCC or CDC respectively. Silation level is very highly related to the functionality or the inflammatory properties of the product. Charge variant is another important property for monoclonal antibodies. So the acidic or basic species may impact the efficacy of the function. Aggregation, fragmentation, again, is also a common issue for therapeutic proteins. The existence of this issue could be problematic because we know it may impact the biological activity and immunogenicity, and may also have some other side effects. So with this one, so it's very important for us to understand the product quality and the productivity differences between the two culture modes. And also at the same time, we're trying to propose potential approaches to addressing these differences. In order to do this work, so we have used 3 different true cell lines, producing protein A, B, C, respectively. Protein A is a glycosylated monoclonal antibody. B is a glycosylated monoclonal antibody. C is a fusion protein. So basically, we're trying to use a wide range of molecules, trying to understand the technical challenges for this transition. For all the 3 molecules here, for the clinical manufacturing, they all use fed batch. So our work is trying to convert the fed batch to the continuous process. So I think you guys may already familiar with the perfusion. So basically, the difference between these two, between the fed batch and the continuous, is continuous can provide consistent nutrient supply, and also at the same time, the product can be consistently harvested through the cell retention device. Here, the cell retention device, we use ATF from Repligen. I believe for John, maybe will provide more in description of this device, so I won't spend too much time on this one. And also, again, for this conversion, so we use the same operating parameters, and also for the medium, so we use basically as a mixture of the fed batch, basal, and feed medium to create our perfusion medium. So so the very important thing for us is to achieve steady state cell growth. In order to achieve this goal, so we use the bleed to control the VCD, and also at the same time we control the consistent cell-specific perfusion rate. With these two well controlled, we can achieve the steady state cell growth profile as shown in the right side for all the three molecules. So basically for protein A and protein C, we can easily achieve 100 million cells per ml, and also the viability can be maintained around 80%. For protein B, we can achieve, we try to control the VCD around 40 million, and the viability can also be well maintained at 80%. For the fed batch for those three molecules, for A and C, they can be easily achieve 15 to 20 million cells, and with viability for protein A is 60%, for protein C is around 90%. However, for protein B, we have some difficulties in reaching high peak cell density. The maximum we can reach is around 5 million cells, with the viability on the harvest day at around 60%. So we have done a lot of development work on this cell line, but it seems the maximum we can reach is around 6 million. But compared to the perfusion, we can easily reach 40 million. So in this case, if we look at all the three molecules, it seems for protein B, there's more motivation for us to put more effort to try to work on the conversion. So this table summarizes the productivity for all the three molecules. So we calculate the annual output. We assume for all the three molecules, the culture for the fed batch is 14 days, and for the continuous culture is 60 days, with 15 days growth phase and 45 days harvest days. As a reactor, we assume the turnaround time is 2 days. So for the annual output for protein A, the 1,500 liter continuous one is equivalent to 1,2,000 liter per year. For protein B, 1,500 is equivalent to 3,2,000. For protein C, 1,500 liter is equivalent to 2,000 batches per year. So please keep in mind, for the perfusion process here, we haven't done any development work. So with more work, maybe the volumetric productivity for perfusion will be much higher than current case. But while looking at all the three molecules, so again, for the protein B, it seems there's more potential and there's more driving force for us to work more and dig more. So this slide shows the comparison between for the specific productivity between fed batch and continuous, with the lines indicating the fed batch minus plus 1 SD. So based on those graphs, we know the steady state specific productivity for perfusion can be achieved. But while looking at the specific productivity for those two culture modes, we notice in perfusion, the specific productivity was consistently lower than the fed batch. So what could be the reason? So as I mentioned earlier, for the medium we use here is a mixture of the fed batch, basal, and the feed medium. So basically, as we know, for the fed batch basal, the major driver is to increase the cell growth. And also for perfusion, the reason we don't have use the commercially available medium, so at that time there's none available. And also, I think now there are also very limited sources for the commercial available perfusion medium. So with the medium we use, it seems we have no difficulties in growing cells, but it's just the cells not growing. So in other words, the cells tend to always go to the growth stage instead of going to a protein synthesis. So now we are thinking whether we can shifting the energy from the growth phase to the protein production and therefore maintain the specific productivity in fed batch. There are multiple strategies. So one is Henry Lin has proposed rebalancing the energy metabolized amino acids to improve QP. For example, the 4 energy amino acids, including the aspartate, glutamate, and glutamate. With those modifications, they can improve the QP. Here, we're trying to thinking maybe we can just limit the nutrients by reducing the perfusion rate. We want to see how that may impact the QP. So we use the protein C as an example. For the open circle is the Cspr. For the solid one is specific productivity. So we decreasing the Cspr from around 30 to 40 picoliter per cell per day to around 20 picoliter per cell per day, and the specific productivity is doubled. So based on this feasibility study, so we know the specific productivity can be increased by limiting the nutrients. And also for the future studies, it could be rebalancing the current media and also remove the redundant components, could be provide a cost-effective approach for continuous. So the next step is to trying to evaluate the perfusion, the 2 culture modes impact on the product quality attributes. So here shows the glycan profile for protein C, which is a fusion protein. Protein B is a glycosylated monoclonal antibody. As I mentioned earlier, protein A is a glycosylated antibody, so it doesn't have glycan profiles. So that's why I only have 2 profiles here. So for the fusion protein, it has multiple N-glycosylation sites. It has very complex glycan structures. So based on the graph, we know for the G1, G0, and G1, in perfusion mode is significantly lower than the fed batch. For G2, G3, G4 in perfusion, it seems consistently higher than the fed batch in perfusion. And there are also slightly higher silation levels in perfusion mode. So basically, the reason for the high silation is maybe due to the, in perfusion, there's a shorter resin time. It can reduce the opportunity of the antibody to be exposed to the endogenous silates, which, for the hydrolysis. So based on this molecule, we know it seems perfusion can promote the more complex glycan patterns. And for protein B, which is a glycosylated antibody, the major glycan forms including G0F, G1F, and G2F. So as compared to fed batch, G0F in perfusion is significantly lower. And also at the same time, and G2F, G1F, the more complex glycan patterns are much higher in perfusion than in fed batch. For both cases, both protein A, protein B, and protein C, we have noticed the same thing. So perfusion can promote the more complex glycan features. So however, we know for the perfusion, for the process change, so we need to maintain comparable glycan profiles. So here we want to use the protein B as an example. We're trying to say whether we can modulate the perfusion glycan profiles to match the profiles to the fed batch by adding process levels. So in order to match those profiles, we're trying to start from the glycan pathway. So based on the glycan pathway, we know the UDP UDP acetyl glucosamine is very important for the G0F and G1F, G2F, those synthesis. So, and also we know, and glucosamine, it can increase G0F and decrease G1F and G2F. So this is a report. This is generally recognized by other people also. So in our work, we're trying to supplement glucosamine in the culture medium, and also we know the glucosamine can be toxic, can be cytotoxic to culture. So we have evaluated different levels of the glucosamine, and we decided to add the lower dose we have evaluated in our perfusion medium. So here shows the results. So without glucosamine addition, we notice the G0F in perfusion is lower than the fed batch. With the glucosamine addition, it increases G0F and decreases G1F and G2F significantly. So basically, with the glucosamine addition, it minimizes the difference between the two culture modes. The similar glycan profiles can be achieved by adding the glucosamine. So in this case, it's feasible to convert fed batch to continuous, even there is a glycan difference. So charge variant is another significant property for monoclonal antibodies. It can impact the in vivo and in vitro properties of the product. So we have here the graph shows the charge variant for protein A and protein B. So in general, for perfusion, can significantly decrease the acidic species, especially in the case of the protein B. So we know that acidic species is highly related to the residence time. And also for the perfusion, the residence time is much shorter than the fed batch. So basically, the perfusion could be an efficient way to reduce the acidic species. But of course, we try to also understand how the acidic species impact the safety and efficacy for this one. So another contributor we evaluate is aggregation. So here the graph shows the aggregation, the high molecular species and the monomer species for all the three molecules. So in general, for protein B and protein C, there's no difference. Generally, there's no difference. But also we can observe the molecular weight, the aggregation level and the monomer level. They have been maintaining consistent across the culture duration. But for protein A, we can notice in perfusion, the high molecular weight is significantly lower than the fed batch. As I mentioned, protein A is aglycosylated monoclonal antibody, and based on the literature, for the aglycosylated antibody, they can aggregate more easily. And also, at the same time, aggregation can also be related to the residence time. In fed batch, as we know, fed batch is much longer than a continuous one. So based on this study, we know the aglycosylated, for in general, the perfusion could be provided as efficient approach to reduce aggregation level if for some antibodies have aggregation issues. So here's some of the summary for today's presentation. So basically, we have evaluated the impact of process change from fed batch to continuous on productivity and product quality for all three molecules. So we have observed lower specific productivity in continuous than in fed batch, and also the differences were observed in glycan profiles, charge, and also aggregation. in this work, we also evaluated that it is feasible to convert fed batch to continuous. So it's feasible to match the specific productivity and also the glycan profiles in fed batch. And also here is the thing. So whether it's worth it to transition it from fed batch to continuous for the late stage legacy or biosimilars. So it's a case-by-case scenarios, actually. It should not just only consider the costs, it also need to consider the company strategies and also consider the technical difficulties. For example, for all the three molecules we have evaluated here, so it seems the protein B has more motivation for us for this transition. So then I would like to acknowledge my previous co-workers who contributed and supported this work. Lastly, I would like to take the questions in the end of the session.
Thank you, Dr. Zhang. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar, but please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now please allow me to introduce our second presenter. John Bonham Carter is a product lead manager, cell culture and clarification business at Repligen. John has a serial entrepreneur with experience in multiple countries in industries from newspapers to biotechnology. Within bioprocessing, he has worked with many different upstream technologies and, most recently, has propelled the XL ATF system to become the industry leader in continuous culture equipment supply. John is a regular international speaker on bioprocessing, SME entrepreneurship, and business coaching. He is the editor of 2 ebooks, Continuous Bioprocessing, Current Practice and Future Potential, and Continuous Bioprocessing, Industry Best Practices. John holds a chemistry degree from the University of Bristol. I'll now be handing over to our second presenter. Welcome, Mr. Bonham Carter.
Thank you, Stephen. Thank you for holding on, everybody. The numbers of viewers keep increasing. That's very nice. Rue, thank you for a lovely presentation on the details of productivity and quality attributes of your previous processes. I'm going to do an introduction here of Repligen Corporation. And I'm going to look at industry trends from a little different aspect than Rue looked at. I'm going to look at perfusion as a lever to impact quality. And I'm also going to lead you through some stepping stones. For those of you who are less experienced in continuous manufacturing, I'm going to give you some ideas about how you can make instant impacts on your process, moving you down that line while looking at the importance of quality in that processing option that you take. So Repligen is a rapidly growing company, in fact, one of the top 100 growing companies in America right now, and from its roots as a protein A manufacturer, has now become an industry leader in hollow fiber and flat sheet filtration technologies, pre-packed chromatography columns, including sterile gamma-radiated chromatography columns, and keeping its core business of protein A manufacture. And every antibody bar one on the market today actually uses protein A manufactured by Repligen, whether you source that resin from GE or other places. So you might know us as a more Repligen inside type of product, but now we are very active in the field with our new products. And you can see this evolution as we've gone through from the early stages in the 80s to the last couple of years as we acquired companies such as Spectrum, Tangenics, and Refine Technology as well, with those associated products and technologies that we've developed. We now have direct representation in 3 continents and expanding rather rapidly in Asia in particular. And we have multiple sites where we manufacture products. We have a very stringent inventory management program across the globe. We support multiple products in commercial facilities across the world and ISO compliant. So let's look at some of the trends. And I think one of the most important things for people to understand here is antibodies are under increasing cost pressure. And that has just not been the case for 20 years or so. And so this cost pressure means that people need to consider that antibodies are going to be like commodities. It may not follow exactly the penicillin life cycle, where I think the two graphs that are missing there unfortunately show that, which is a dramatic drop. But for example, AbbVie has recently reduced the price of Humira by 80% in some locations. So this cost pressure and the competition of different molecules for a same disease indication are having impact in process engineering and modes of manufacture. Now, manufacturing is becoming a competitive advantage, and it's important for companies to appreciate their skill levels here and how they make investments. And some companies, such as Just Bio and others, have clear targets of saying this needs to be at $10 a gram or less. And of course, before, with the margins being high, the cost of goods has not been important. What's been important is first to market and the ability to control that market and covering the R&D costs. That is shifting. So the move from fed batch to continuous is absolutely going to happen at one pace or another, but it's being driven by cost pressures as well as technical efficiencies. So here are the graphs where you look at where might biotech and pharma be and saying, why are we different to pharma? Well, biotech's a little earlier in the stage. Antibodies may be a little further along, but gene therapy and cell therapy even further to the left. And then when we look at that comparison to penicillin prices, where might biotech prices be? Well, we're right at the beginning of this cost. So when we think of us, we've really got to think of our industry being really quite immature in where we are, and we're at that process of industrialization and scale out, essentially, of manufacturing techniques. So the product I've been involved with for about 20 years now is actually this ATF system, which is, I wouldn't say a radical design, it's a very simple design, keeping a filter cleaner than you otherwise expect, and making it simple for an unexperienced person to run perfusion. And why are people interested in this? Well, I took a paper published last year by Jean-Marc Bilsa in 2018, who just reviewed a few papers to say, well, what's the industry standard? So if you're looking to compare yourself, you could look at these papers here and say, what kind of cell densities are being typically produced here? And you can see the top one, 50 million cells, using an online biomass sensor, like an Aber or a Hamilton VCD probe, and then looking at your CSPR levels for this first one, and you can see the variation getting up to 100 million cells, but a lot of it around that 40 to 60 million cells mark, right? At somewhere between 20 and 70 picoliters per cell per day, right? So these are very average. Important to point out that many of these authors are academic and not necessarily with the best cell lines in the industry. But certainly this is a reasonable stake in the ground that you could look at and compare yourself to. If you were to look previously, 15, 20 years ago, the retention device would not be ATF or TFF. It might be a cell settler, it might be a centrifuge. There are many other methods that people have utilized over the years to achieve the same goal, but it's just ATF has become the most popular device for a multitude of reasons. When we look at some other data, and I've got some graphs here, you can see where maybe the state of the art is going. So top left, we have a university, Stockholm University, pushing it up to 200 million cells per milliliter in a relatively steady state until that last day. Top right, we have insect cells hitting 350 million cells per milliliter. Bottom left, the CHO cell from an East Coast biopharm company, taking a fed batch process, intensifying it. It's not a continuous culture experiment. It's an intensified process being run there. Again, 10 times the cell density, up to 300 million cells per milliliter. And bottom right, data from Repligen comparing single-use and stainless steel at about the 80 to 100 million cells per milliliter. So when you look at this, you realize very clearly if you can control the media correctly and get that cost under control, the productivity gain is going to be significant, right? It is not just a question of 10 or 20% improvement. It's a question of, as Sanofi presented last year, 100-fold improvement in productivity in some processes. That is a real driving factor behind these changes towards continuous. And what does it mean? It allows you, as Ru indicated, to make significant savings on capital. So when you're going to build a new facility, instead of spending $1 billion on a 20,000 liter facility, you might spend $100 to $200 million on a smaller facility because of savings in HVAC, savings in clean room space, savings in stainless steel, and that facility will have a higher capacity of operation, and you'll be able to more flexibly respond in demand and build extensions as market demand becomes known, or as different products mature in their market. You'll be able to manage that risk and that portfolio better. But it also means you've got R&D scale as commercial scale. So you eliminate scale up, eliminate that risk, and save a lot of time and cost while delaying that decision to build. So each of these things you've got to think of is, it's not just the cell culture performance and the productivity of that, it's the whole business and how you impact that risk profile of the business. So cell culture upstream scientists might get excited about their cell density, but the business decision is a little wider than that, and that's got to be taken into account. So the FDA has been incredibly positive on this move towards continuous. It's worth noting that continuous processes upstream have been around for 30 years, so there's nothing new in how you define a batch or how you make exceptions to batches when there's a quality issue you need to deal with. But that knowledge has been in the hands of a very small number of people, and now it's getting wider. But it's very important to understand there's nothing being invented here on the regulatory regime of continuous upstream manufacture. The FDA more recently, a couple of years ago, took this slide from Rapti Meduri to say, well, how would you go about thinking about this shift in mindset from fed batch to continuous? What are the kind of things you need to look at? There's science and engineering concepts, there's risk, operational issues, strategy of control, and experience and maturity. Each of these bullet points look at things that you need to understand, you need to characterize, and you need to appreciate how your organization is going to solve each of these items. And knowing that there are answers out there, whether from consultants, employing people, or working with CMOs who have all of these experiences. But each of these items does need a shift in thinking. So you should not think this is a nice, easy, straight on, we'll do continuous and everything falls into place. There's investment, and there's work, and there's time to get right as you make this shift and understand those risks. And importantly, training is something that I really need to emphasize as you make this shift towards continuous. And a company like Repligen, of course, is there, and we provide free training as you understand each of these changes from fed batch to continuous. So one aspect to look at is this quality, right? A key aspect is if I'm going from fed batch and I'm going to continuous, what is the impact on my product quality? And I still think, this goes back several years, I still think this is the best data I've seen to clearly explain this. This is ETH Zurich University, Professor Massimo Morbidelli, who many of you might know from continuous downstream operation, continuous chromatography. His group here has produced data that when you see here bottom right, the viable cell density data, starting for sort of 7-8 days and 20 million cells, they then shift the metabolism up to 60 million cells for about another 7 or 8 days, and then they shift it down to about 40 million cells for another 7 or 8 days. And they do this by different bleeding rates and different feed rates. So within this 1 month run, you've essentially got 3 different experiments and looking at quality and productivity in each of those steady states and saying, what is the impact on my process? Where potentially should I be operating my process for maximum productivity and the best product profile I'm trying to do? And you can see here on the top right how the titer, of course, changes. Unsurprisingly, more cells means more product. And in theory, that would be exactly where you want to go. But it's important to understand, and you can see this in bottom left, that the product profile changes according to the cell density. Now, of course, once you've got control of this, and you can see the green, kind of pinky red slide, pinky red color and the blue color map to the green, blue, and red stages in this VCD graph, you're going to say, where do I need my process to be? But Perfusion is giving you that lever into saying, I can control my process. I can move from one state to another. I can do R&D and further optimize and improve the quality of that, and then keep it steady for the whole process. Of course, in fed batch, you've got a different process every single day that you're trying to manage. And you end up with a heterogeneous mixture that then goes in one batch downstream. So Perfusion gives you that lever and that control of quality and that tightness that fed batch doesn't, but it means you need to understand how to use that lever. And that takes time in R&D to achieve that. So another aspect here is to say, okay, well, when you're running an integrated continuous process and you've got this reactor here, and I'm using an example of reactor pH here, if I've got a continuous flow through my system and a surge bag going on to continuous chromatography, what happens if that pH line moves from the set point down into a lower lung, right? Is that meaning I need to reject that product? So is this a question of, is this a critical product to quality attribute that now the product quality needs to be rejected? Or is it something which has an impact on column performance? Or is it just a productivity issue? You're going to produce less of the product at that pH. You need to have characterized this, understand it, and see how it fits into your whole process control strategy. Maybe you need to separate it out from the continuous flow to reject it. You need to understand the residence time of calculation, the mixing in your facility and that of that process line, so you can make that separation. But if it's just operational and productivity issue, then that might be simpler. But you still need to say, well, can I bring that back into control? So the surge bag, when it passes on to chromatography, it's still the same pH that I constantly want so my columns are operated in the way that I like. So you need an upstream to be thinking about how this works in downstream, an integrated team approach. And this is exactly how people such as Merck and Sanofi Genzyme work in their teams today. The upstream and downstream people are not throwing things over the wall to each other, they're working as integrated teams. And it takes time to understand how this performance can occur as you move a process through. And of course, this is relatively complex. These companies have invested a huge amount of time. So let's think about what can you do today in small steps in your organization to say, can I get some of those benefits without having to massively change your organization from fed batch to continuous, right? That should be maybe a goal in 5 years, but can you do something in the in-between times to get some of the benefits and productivity of continuous without having a huge regulatory impact and a huge philosophical change in your company, right? And so, of course, this concept of moving from fed batch to continuous concentrated perfusion is what people have been doing. That's what Bruce spoke about. That's what some of my slides earlier are talking about. And people such as Sanofi, when you compare with the Genentech data Ru said about 10 to 15 grams per liter titer, Sanofi published last year about 4.5 grams per liter per day. So when you've got the experts running their high productivity processes, you can deliver significant grams per liter coming out every single day, right? But the average is about 60 to 100 million cells, 1 to 2 grams per liter per day is what we would expect people to do, right? So that's your real benchmark to take into your companies and compare with. So the intensification options that I would suggest you could do as a stepping stone is to take your existing fed batch processes and choose one of these two, or both of these processes, N-1 perfusion or high productivity harvest. And I'll go into each of these in some detail now to see what's the benefits and costs of each of these that you do. The key to them both is saying there's a single harvest, a single QC point is maintained. So it's a very small shift in mindset, some gains, but you can retrofit this to existing fed batch facilities and understand the impact on your quality of process. So why are people doing this? Here's some data from a collaborator here showing in blue a fed batch control, a previous process, and then when they've inoculated their final stage bioreactor at a higher cell density at 10 million cells here, you can see a similar growth rate, but a peak cell density which is higher, and then a slowdown of a similar amount, a similar death rate here. But it means that the fed batch titer in the final bioreactor is reached, the same titer is reached a little earlier. So you're going to save time in that production bioreactor, which turns it over quicker, which means in that same facility, you can get 30 to 40% increase in throughput, but on a same fed batch process, right? So a defined change, a change you can characterize, a change that multiple companies on legacy processes have already implemented, and many biosimilar companies have already implemented. So the risk here is low, and a defined method of taking this forward has been approved and worked on by many companies. So it's a really nice way, and the perfusion, to get this higher cell density in the previous reactor. You have to assess that impact on quality. It could be there, but it's not in your final bioreactor in terms of how you assess that performance. So if we look at the clarification side, the high productivity harvest process, then we're trying to say, can we eliminate clarification and simplify that unit operation while gaining productivity? There's very few people I know that, I can't see you, but if you put up your hand and say you love working with a centrifuge, I'm not really going to believe you on that one. Most people don't like it. It's a complex step, but it works. It's understood. The question is, can it be improved upon, right? Can depth filtration and the extreme surface areas needed and the variation from fed batch colleagues thrown over the shelf as you deal with that variation in primary recovery, that's also an issue, right? So we're trying to say, can we improve upon that? And we would argue that you can eliminate those two steps, replace it with a alternating tangential flow filter, keep that filter clean, and perform and harvest straight from the reactor, going onto a column with maybe a sterile filtration unit as a intervening step. And it's significantly simpler. But what does it mean in terms of operation, and what is really the gain? Can you remake this elimination? And what is your productivity gain and impact on product quality? So first of all, what do we mean by this process? What we mean is you run for 10 days your standard fed batch, and then you say, okay, for the next 4 days, I'm going to attach a filtration device here and start diafiltering and start harvesting the material inside my bioreactor. So this might be as little, if this is a 1,000-liter bioreactor, let's say, this might be 100 liters a day that you are diafiltering through this, and you're harvesting that 100 liters a day, right? It could be as much as 1,000 liters a day, but a minimum of 100. And you do that for 4 days, and at the end of 4 days, you get to your standard day that you would typically harvest upon, and then you would start to draw down that remaining material in the bioreactor. So you're basically turning off that feed pump going in and drawing down the harvest. And now you've speeded this up, so it's 1 to 2 vessel volumes a day, meaning 1,000 to 2,000 liters a day if you've got a 1,000-liter bioreactor. And as you draw this down and harvest it out, you're going to get to a minimum working volume and recover everything, and that's going to be the end of your 14 days. So on Friday evening, you will have achieved your harvest of the reactor, and this is now a clarified batch that you can move onto your column. But the nice thing about it is, what was the work involved in doing that? There's no buffer prep, there's no centrifugation, there's no rush to get that done on Friday morning to get home Friday evening, right? This is literally turning on or off a pump and harvesting it out in a very reliable manner on a device that's used thousands of places worldwide today. So highly predictable, easy to scale down, and let's look at some of the performance of this to say, well, what's the end result? So in this scenario, we've run 4 experiments here, and we've started high productivity harvest on day 10, as our little cartoon showed, and we've done at 3 different flow rates here, 3 different harvest rates, quarter vessel volume a day, half and 1 vessel volume a day. And you can see the impact on cell density, of course, as you have these different flow rates. You're removing material, you're avoiding cell death, so of course cells are going to react better in this way. And you can see you can have quite an impact at some of the high flow rates. And that may or may not be appropriate for your process, but it's an interesting result to know what is possible. But the key about this is not just the amount of cells that are being produced here, but the viability. You see the change in viability. Instead of viability dropping, viability gets maintained and in fact increased. So the impact on product quality here could be, for some processes, rather significant. Instead of producing host cell proteins, instead of producing species which cause aggregation and changes in glycosylation, which are all negative, you might be able to maintain it. When you look at the cell-specific productivity, something that Ru touched on as well to look at, what's happening here in the effect on the cells, you can see as the filtration rate goes up, in fact the cells respond in a more positive way. So it goes from 4.6, and with each increase, the cell-specific productivity goes up. So the cells are actually in a higher productive state. They're in a better environment, producing more protein that's being harvested. And then when you look at the assessment of of the total accumulated protein at the end of this process, remember we started on about day 10 here. When we compare with fed batch, this 1x, which is about 800 mg per mil in our particular cell line, we're getting a 20% increase at a 0.25 VVD. We're getting a 1.4 increase, a 40% increase, and then a 100% increase when we're actually doing it at a very fast flow rate, right? Now, there's very few people I've met who've got higher than 100% yield on their harvest. And each of these processes is providing higher than 100% yield. That's a very nice number to take through to your senior management to beat 100% yield. Of course, there's a cost, right? And the cost is the media, right? So how would you determine what is the correct cost and options for choosing? So we can first say, well, let's use the most amount of media to gain the highest amount of productivity. That might be a great choice if you need to produce a lot of material for research or in a phase 1 process to be able to say, I just want one reactor run, and instead of running 2, I'm going to run 1 reactor in the same period of time, and my clinical facility can turn over much quicker, get more molecules through that facility. That's nice and easy. But maybe it's a different. Maybe you're slightly later in the process of your clinical manufacture, and you have less room for maneuver. And this little arrow that seems to have been a little shifted here should be saying from day 14, the same productivity on the red line matches day 11 of the blue line, meaning if you do 1 day at 1 vessel volume a day of harvest, then you've actually matched the same productivity as you would have done in 14 days. So if you're looking to say, my window of opportunity, I'm say post phase 2 and I'm looking for some improvement, I'll take my 3 days of time saving and I'll turn that reactor over quicker, but that same batch will be passed downstream with a minimum of change to our existing clinical trial material already produced. So that might be the option for you to look at and say, is that the way I should be moving forward for this particular process? So depending on where you are in your clinical process, you might make different decisions. If you're trying to make a platform process, you've actually got a lot of choices here about how to operate to gain either flexibility, productivity, or get control over product quality as you remove the byproducts and species you would not prefer to have in your reactor. And then let's look at some metabolite data. So here we're overlaying data from glucose, lactate, and ammonia. You can see glucose pretty well matching. In lactate, you see, of course, the shift on day 10 to a growth in the orange and the blue graphs here. That may be a problem in some people's processes. It may not be a problem in others. You can see the green at the lowest flow rate matches pretty well the previous red. That's going to be closer to where maybe you want to be in a later stage process. And you can also see in ammonia. Of course, on the red line, the old fed batch, this is actually as cells are dying, ammonia has been created. This could be a problem downstream, right? And that problem is avoided in most of the high productivity harvest processes going forward. So of course, behind this, you've got to say, well, what is the actual glycosylation profile, the aggregation, the host cell protein data? You're going to be overlaying that and say, how should I be optimizing my particular feed rates and harvest rates for my particular process? And when we look at the one piece of data I can share with you today on host cell proteins, when we look at this, this is pre-purification. So this is prior to the depth filter and prior to centrifugation. We're saying on the traditional fed batch, we're up at sort of 150-ish. But on the high productivity harvest, it's not that we're filtering out this material. It's that it's not being produced in the first place. The cells aren't dying, so host cell proteins aren't being released, and you're not having to filter it out. So rather than clear up the mess, we're not producing the mess in the first place. So this is one of the levers you can use in your R&D to try and manipulate that process to make a higher quality product. Of course, if you wanted to have the best, you're going to move to a long stage, steady state, high intensity, continuous process. But these are the steady states that take a huge amount of investment, and this is just a stepping stone to get you towards there and make it easier. So in summary here, when you look at high productivity harvest, we're looking to say, can you eliminate that recovery and clarification step you previously had? Move it to a process where you get increased productivity or save time, certainly save hassle for your teams, but also get this healthier culture, lower process-related impurities, simplicity of operation, scale down models that are reliable and have a closed, single-step process, sterilized produced clarified media that can go onto a column, right? Ready straight for purification. And to remind you on that N-1, again, another way of keeping a single QC point, a single harvest, to be able to simply retrofit facilities on that stepping stone as you move towards a continuous operation further down the line when your organization is ready. And with that, I'd like to finish, and I think Ru and I will be very willing to take any questions.
Thank you, Mr. Bonham Carter. And now, as he just mentioned, we will begin the question and answer session of the webinar. But as a reminder to the audience, you can still send your questions in via the questions tab located directly below your webinar screen. So now, thank you. Our first question is, in addition to technical challenges from your perspective, what are the regulatory concerns, challenges, while converting fed batch to continuous manufacturing? Ru, I believe this question is for you.
So, okay, thanks for the question. I think it's a pretty good question. So actually in John's presentation, so he has a slide to talk about the general regulatory concerns. You know, basically in general thing is there's no regulatory guidelines or requirements for continuous, basically. But from a little bit more specific thing is kind of like, I would think the challenge is like a segregation of the non-conformance materials. For example, here I'm only talking about the continuous upstream. For one upstream, it may have multiple harvest slices. If one harvest slice is out of specification, what could we do? So there's no for sure answer for this one. So definitely it's very important for the company to work with FDA to figure out a solution. And also another thing is for the changing processes, there's no specific guideline for the continuous one, but there's a guideline for the significant process change. What you could do is, we have to do the comprehensive comparability analysis, try to demonstrate process change will not change the PQAs for the safety and or efficacy. I think that's like some of the regulatory concerns I would think of right now.
Yeah, I think I'd like to add to that, Ru, as well, to say this is a key question as people try and assess, how much work is there involved in that transition and what risk is there and how ready is my organization. But actually, this is a well-trodden path, even if your organization is only just starting down that path. And its simplicity, it's everything that Ru said, but in some respects, it's simple too. It's instead of having a 14-day window where you produce 2,000 liters in that 14 days, or maybe it's 20,000 liters, but if it's 2,000 liters and a 2,000 liter bioreactor in 14 days, now you might have 4,000 to 6,000 liters every 3 days as a batch. But the key is you have to show that each batch is under control. Whether that's a fed batch of 14 days or a continuous process of 3 days, that 3 days is the same as the next 3 days, is the same as the next 3 days. And if you're showing that with a QC and the understanding of the process control and everything's under control, you've achieved your objective. And the problem shifts to how you would operate that facility rather than any regulatory hurdle, right? The regulatory hurdle is very easy to understand. It's just difficult for companies to make that shift without that experience.
Hey, John, thanks for the comments. I agree with you. I think maintaining consistent product quality is definitely the advantage for the continuous one. So I think definitely with the quality under control, for my concern, I think could be addressed, I would say.
Our next question is, we have an existing fed batch process in phase 1. When would you consider it appropriate to change to a continuous process, if at all? I believe this is for you, Mr. Bonham Carter.
Thanks. Yeah, well, of course, I'd love to say right now, right? You can give me a call and I'll help you. But I think the answer is strategic as well as technical. The key is, why would you change at all? I think the question kind of states that. I think you have to understand what in your company is requiring you to change. Is it that you do not have a facility at large scale and you've got an uncertain market demand, right? Is your company ready to either contract out with a large company or try and build their own facility? And when you start to answer that question and you start to look at the market demand for your molecule, then you start to say, well, what are the business drivers behind doing my own facility, contracting out? What's the demand? How am I going to manage that? A continuous process allows you a little bit of a delay in that decision. It allows you to scale out if you get that market demand incorrect and you need to rapidly increase productivity, and it allows you to put less money down up front as you develop that process to enable, if you've got a smaller market demand than you thought, and you haven't wasted capital expenditure. So really, it's a strategic question rather than a technical question. If you wanted to look at just the really specific technical part of it is, well, is your product quality sufficient in fed batch? Would you gain by moving it to continuous? Do you get a better product control? Is there a clinical effect of that? That might be the driver instead. But typically on a MAB, it's going to be more driven by business.
Thank you for that. Our next question is, what are the major technical considerations in order to successfully convert fed batch to continuous processing? It's for you, Dr. Zang. Do you want to answer first?
Mm-hmm. I think there are some major concerns, definitely. I would say the starting point could be the medium itself. Because currently, for the commercially available medium for the perfusion, I believe it's only one manufacturer, which is Sigma. So I think for majority of the pharmaceutical companies, they either develop their in-house medium or they can create the perfusion medium from the fed batch, basal, and feed. So those ones not really specifically developed for perfusion. So there are some challenges. And also the second concern is for perfusion, usually the goal is to achieve steady state high cell density. So with high cell density, which means there are high gas demand, and also then the share could be a concern. So in order to make sure for the conversion to be successfully, you have to well consider the spatula design, gassing strategy, and also how to protect the cells. What's the mitigation strategy for the share? I think those are the things very critical for achieving high cell density culture. And the third factor for process the challenge is how to match product quality in perfusion to the fed batch. Because based on my presentation, we know definitely there are some differences, product quality differences. So how can we modulate those ones to address the regulatory concerns? I think those are the three of the most important technical considerations for a successful transition from fed batch to perfusion.
Thank you. Our next question is, do you believe that an optimization of the perfusion media can have a critical impact on the increase of productivity? Mr. Bonham Carter, would you like to take this first?
Yeah, I'll give a quick answer, and Rue might want to give a more detailed answer. Yes is the simple answer. It is an utterly critical element in terms of both cost and quality to look at perfusion media. Many people take off-the-shelf media in preclinical and early preclinical, but as you move later, it is absolutely expected that you would do some optimization to both reduce that cost. It might be more dilute media, it might be more concentrated media, depending on what you're trying to achieve. But I would expect everyone to look at optimizing the media.
And Ru, do you have anything to add? Do you believe that an optimization of the perfusion media can have a critical impact on the increase of productivity?
Yeah, I do think it's very critical. I think as for my answer to the technical considerations for the conversion, I also list the perfusion medium optimization as a very critical consideration, because based on my experiences, cells can easily achieve very high cell density, but sometimes for high cell density, 80 million cells and 120 million cells, the productivity are very similar. So which means the medium is not really balanced. The cells just keep growing, but they are not producing. So definitely rebalancing, in addition to the cost, John just mentioned, I would say rebalancing the medium, trying to shift the energy from the cell growth to the production. That's very critical. So I would say definitely yes.
Thank you. Our next question is, do you generally use the same Cspr for each process? If not, how do you develop an appropriate Cspr rate? Ru, would you like to take this first?
Yeah, for the Cspr, actually the Cspr from my presentation, so I use the very platformized, cell-specific perfusion rate based on my experiences. So I think the minimum requirement for Cspr, you're trying to make sure there's no nutrients limitation. So based on my experiences with the commercial medium and also perfusion processes, we have a starting point. But definitely, we use that as a starting point. It doesn't say, it's not changing over during the process development. For example, for each cell line, so we use the same starting point for Cspr, but at the same time, we do the spent medium analysis, trying to understand cell-specific consumption and decide which one to go for the final Cspr. So that's my answer.
Yeah, and I think that's a very good way of answering the question. I think if the questioner wants to look at the Repligen website, there's a paper by Veronique Chateau in a book we published on continuous bioprocessing, where it gives a step-by-step procedure of how to develop a Cspr for a new process. So a starting point and a series of steps of how to optimize that. And obviously it's a guideline that you can improve, but that's where I'd point you to answer that question.
Thank you. Our last question is, doesn't it take longer to develop a process in continuous mode than fed batch? John, would you like to take this question first?
Yeah, I think the perception from everyone is because it's a 30-day process or a 60-day process rather than a 14-day process, my R&D time is extended proportionally. And I think, when you first start out, that's probably true. But when you get to a certain level of sophistication, that is not true. As I showed in one of my slides, in that 30-day process, you can do 3 different experiments, different cell densities to understand the impact of cell density and media on product quality and productivity. So I think as you get better at understanding how to use the tool, so you will improve your timeline. But right at the beginning, yes, you're going to have to learn a whole set of new tools like learning to drive, learning to ride a bike. There's a learning curve which will slow you down until you've got a level of expertise that you can then speed up again.
So I have some comments to add to John's comments, actually. So based on my experiences, I would say, maybe it's really like based on experiences, for development timeline for the perfusion could be longer than the fed batch process. As John mentioned, the duration could be 30 days versus 14 days, and it should be that's one thing. And also, if let's say if we can really truly develop a very consistent continuous process, which means it can deliver consistent product quality attributes and consistent VCD or viability profile, everything is consistent. And at that time, I think that can go back to John's thing. Basically, you can use one run to study different conditions, 3 or 4 conditions that could be shorten the timeline. But I would say it's very challenging to really develop a continuous process that has very consistent quality attributes.
Well, thank you both for your answers, and thank you, audience, for all of your questions. Unfortunately, that's all the questions we do have time for. But I would like to take this opportunity to thank once again our presenters, Ru Zang, Associate Director of Mersana Therapeutics, and John Bonham Carter, Product Line Lead of Cell Culture and Clarification Business at Repligen, for sharing their knowledge with us. I would like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll try to get those answers to you. Thank you, everyone. Have a great day.